Okay, today we're going to go over the RNA interactive notes. We've talked a lot about DNA, so we're going to start comparing DNA and RNA. So the sugar in DNA is deoxyribonucleic acid. So the sugar in DNA is deoxyribose. And we talked about DNA being a double helix. It looks like a twisted ladder. So there's two strands. There's that right half. This is complementary to the left half. So there's two strands in the DNA. We have two sugar phosphate sides with their bases connected to another base that make the rungs. And then the other side there. So two strands of DNA. And the bases in DNA, that's the adenine. Adenine bonds with thymine. Then we have cytosine that binds with guanine. So that's DNA. That's what we've been talking about for this whole week. So we're familiar with that. So RNA. RNA is ribonucleic acid. So the sugar in RNA is ribose. So we have deoxyribose in DNA. We have ribose in RNA. And RNA is a single strand. It's like half the ladder. I mean, we talked about DNA helicase coming in and unzipping, and we looked at that when we talked about DNA replication. And it would split that DNA molecule, that twisted ladder up the middle. Well, RNA is just like one half of that DNA. So it's a single strand. The bases for RNA, we still have adenine. So we still have the A. But in RNA, thymine is replaced by uracil. So I'm going to go ahead and write that whole thing down since we haven't talked about it. Instead of T's, we have U's, uracil. And then we still have cytosine that binds with guanine. So we still have our adenine, our AC's and G's, but instead of T's that we had in DNA, we have U's. So there will be no T's in our RNA. If it's something where you think you should be putting a T, it's going to be a U. Okay, so uracil takes the place of thymine in RNA. So what does RNA stand for? Well, we just said the sugar in RNA is ribose. And we said the sugar in DNA was deoxyribose. So DNA was deoxyribonucleic acid. Unless it's the sugar in RNA is ribose, then it's just going to be ribonucleic acid. Ribonucleic acid. Okay, number 3 says, what type of macromolecule is RNA? Ribonucleic acid. Remember back to our macromolecule unit, nucleic acid is one of our macromolecules. So that is what RNA would be. RNA is a nucleic acid. It is made up of nucleotides. Nucleotides are the building blocks of our nucleic acid. So it is a nucleic acid. Number 4 says the process of using DNA to produce complementary mRNA strand is called, okay, we're going from DNA to mRNA. And mRNA, I call that, that's our messenger RNA. It brings that message. The DNA is in the nucleus, remember, and it can't leave the nucleus, but it has the instructions for making proteins. Okay. Who's making proteins, remember? Proteins are made by the ribosomes, and the ribosomes are either floating around out in the cytoplasm or they're going to be attached to the rough ER that's out there in the cytoplasm. They're outside the nucleus. So you think, what good is the instructions for making proteins if it's stuck in the nucleus and the ribosomes that are going to be making the proteins are outside the nucleus? Well, that's the issue. DNA cannot leave the nucleus, but our ribosomes that make those proteins around the nucleus. So mRNA is what's going to take that message from the DNA in the nucleus out to those ribosomes that are out of the cytoplasm. Okay, so mRNA is going to take that message, those instructions for making proteins, from the DNA outside the nucleus to those ribosomes. So So mRNA, M is messenger. It's carrying that message that has those instructions for making the proteins. So it says the process of using DNA to produce the complementary mRNA strand is called, it's kind of like when they go in, kind of like when we talked about DNA replication and it copies. Very similar, but this time we're making the RNA, and it's going to copy the specific genes for making the protein of interest at that time. And it's going to go in and it's going to lay down those complementary bases, much like we talked about in DNA replication. But this time it's just making a single strand of that mRNA. Remember, RNA is going to be single-stranded, and there's no thymine. It's going to be laying down uracils where we would think we need a T. Okay, so that is called transcription. You transcribe mRNA from the DNA to transcription. Transcription is the process of going from DNA to mRNA. Number 5 says the sequence of the blank in the mRNA complements the sequence in the DNA template. So it's the sequence of the bases. The bases are going to be complementary to that DNA strand. This is the sequence of the bases. That's your A's and G's and C's. In the case of mRNA, your U's. The sequences of bases in the mRNA complement the sequence in the DNA template. That means A's are bonding with, this time, U, and G's and C's are still together. In eukaryotes, U means true. U means they do have a nucleus. So in eukaryotes, mRNA is formed in the blank and then travels to the blank. Okay, we just said it has to be copied from the DNA. DNA cannot leave the nucleus. So mRNA is formed in the nucleus. And then it travels to the, has to go out to the cytoplasm where the ribosomes are. So we're going to put, it travels to the ribosomes. The cytoplasm. Some of them are floating around. Some of them are floating around free in the cytoplasm, and then some of them are going to be attached to that rough ER, remember. The rough ER is also out there at the cytoplasm. So it's going to go, it's made in the nucleus. It's going to go through those nuclear pores and the nuclear membrane out to the cytoplasm where it can find a ribosome attached to the rough ER there or out in the cytoplasm. So the enzyme blank binds to DNA during transcription to make mRNA. You might remember that we talked about DNA polymerase when we talked about DNA replication. We said the helicase comes in and unzips it, and then the polymerase comes in and puts down the D bases, matches up those complementary bases, puts down and proofreads. So if we're doing RNA, it's not going to be DNA polymerase this time, but it is going to be RNA polymerase. RNA polymerase. Again, it's going to be putting down and proofreading. This time it's making RNA. So it'll be putting, if there's an A on our DNA template, it'll put down a U. It'll put down a uracil this time. If there's a G, it's going to put a C. If it's C, it's going to put a G. If it's a T, it's going to put an A. T's are still going to go to A's. But if it's an A, there are no T's in the RNA, so it'll be putting down a U. But polymerase puts in and proofreads. Remember, it starts, they all start with a P. Polymerase puts in and proofreads. Number 8 says RNA polymerase binds to a region of the DNA called blank, which are start signals for transcription. And so special regions on the DNA molecule that are going to trigger it to start making an RNA molecule, those are called promoters. Yes, that's promoters. P-R-O-M-O-T-E-R-S. Promoters. Promoters are going to signal when to start making that RNA, the mRNA molecule, and then there'll be another place that'll tell it to stop making it. So this is all you need to stop making it right there. Okay. It's also going to go through a series of what's called RNA splicing, where it's going to take out part of it that doesn't actually code for that protein, and it's going to keep certain parts that do code for the protein that it's making, and it's going to stick those back together. So it does go through a process. Once that mRNA is made, then it is processed to code specifically for the needed protein. It's going to go out that nuclear pore. It's going to go to a ribosome, and then at that ribosome, it's going to be translated. That ribosome proteins, they don't understand the DNA language, so it has to be translated at this point. It's bringing mRNA, it's a messenger, bringing that message from the DNA written in DNA language. The mRNA was just transcribed from the DNA. That's like taking down notes there, transcribing. But now it's got to be translated into a protein. So that's going to happen. Translation is the coding of the mRNA into a protein. That's going to happen at the ribosome. So the ribosome is going to do that, translating it into a protein. Into a protein. So protein is our blank there. And there's those two steps. There's transcription, which is the mRNA being made from the DNA, and then it goes out to the ribosome where it's translated into a protein. And they happen in alphabetical order. Transcription alphabetically comes before translation. So transcription is the first step, happens in the nucleus. DNA is transcripted into mRNA. And then it gets to the ribosome has to translate that DNA language into protein language. And that happens at the ribosome. So where in the cell does translation occur? It occurs at the ribosome. Ribosomes make proteins, and those proteins are made by translating that mRNA into protein language. Okay. There's actually other steps, and I'll list those for you in just a second. So use the base pairing rules to identify the bases on the mRNA strand that are transcribed from the following DNA template strand. Okay, so if it's an A, if we're making mRNA, let's see, I need to go where I need to go. If it's making an RNA, A, we used to say A goes to T's, but RNAs don't have T's. So if it's an A, it's going to be a U. G still goes to C, C still goes to G. Okay, T still goes to A. C goes to G, C goes to G. A goes to U. G goes to C. And A goes to U. That's what that would look like. So this is our DNA template. This is our mRNA strand. And I'm going to put that up there. That's our mRNA. And then number 12 says identify the DNA template that this mRNA strand came from. So this is the mRNA right here. And this time we're going to do the template. We're going to go to the DNA. So U goes to A, which we use like T's. So U goes to A, G goes to C, and C goes to G. Okay, A, we're making DNA this time, so we still have our T's. A goes to T, C goes to G, A goes to T, C goes to G, G goes to C, and U goes to A. And that's for DNA. If it has a T in it, it's DNA. If it has a U in it, it's RNA. And we've talked about messenger RNA. Messenger RNA is the one that's bringing the message from the DNA to the ribosome, that message of how to make those proteins. And we'll talk a little later about tRNA. tRNA, I call it the tow truck RNA. But it's actually the translator RNA. It's the one that's going to actually be picking up those amino acids. And these are going to be codes. Remember we talked about triplets and codons. Each of those three bases of an mRNA is a codon that's going to code for a specific amino acid. And the tRNA is going to go pick those up. And we're going to talk more about that. That's cool stuff. So we'll talk about that later.